We can use electrodes for electric stimulation of tissue, and in this situation we have two types of stimulation and two types of reactions. The first one is constant current stimulation. As you can see, we apply a constant current on the tissue, and the response is as a voltage. With rising edge of the current, based on the ohmic characteristic of the tissue, we will the voltage will increase suddenly with the current. But because of the DC part of the constant current stimulation, the charge inside the tissue will move. Therefore, we have a polarization part of a voltage. We have a increasing part of voltage, and at the end of the simulation pulse, the falling part, we have a voltage difference as the rising part, and after that a repolarization part again here because of the charge moving. Remember that constant current means we need charge to maintain, to preserve the voltage as a fixed value. And in the second type of the stimulation, we can apply voltage constant. And as you can see, when the voltage is constant, also we have a current rising suddenly by the rising edge of the voltage, and also a falling part at the end of the simulation. But because of the voltage fixation, voltage DC part, because the voltage, the stability of voltage needs to fixation of the charge and fixation or preserve the charge at the two sides of the, for example, the cell membrane, leads to a deficit of electric charge. Therefore, the current will be reduced. And you can see a polarization in the current, as you can see here and here. And this is the difference between the voltage and current stimulation. We should consider some points. Simulating electrodes cannot be modeled as a series resistance and capacitance. There is no single useful model. Also, the body electrode has a highly nonlinear response to stimulation, and large currents can cause cavitation, cause cell damage, and heating also. And in platinum electrodes, we use for stimulation. Application is for neural stimulation, and steel electrodes for pacemakers. In this slide, as the last slide of this lecture, we compare some types of electrodes and its applications. As you can see, external electrodes can be used in human level and animal level as an external method for recording signals. But subdural electrode is used in human level and also in animal level with some invasive surgical operations. We should use some surgical and operations to insert these subdural electrodes inside the body. Microelectrodes is a method for deeper recording of data in also human level, animal level, and tissue slice, and also cellular level, as you can see. But microsensors isn't used in human level, and microsensors are commonly used in animal level, tissue level, and cellular level. When we are talking about human level and animal level, we are talking about in vivo application. We are recording when we are recording from a live system. We are recording in vivo, and in vitro means that we isolate some tissue or some part of an organ, and we record the signals in the lab. We called it in vitro applications. Okay, thank you for your attention.